So concept number 5. Mitochondria and chloroplasts change energy from one form to another. Organisms transform the energy they acquire from their surroundings. In eukaryotic cells, mitochondria and chloroplasts are the organelles that convert energy to forms that cells can use for work. Mitochondria are the sites of cellular respiration, the metabolic process that uses oxygen to drive the generation of ATP by extracting energy from sugars, fats, and other fuels. Chloroplasts, found in plants and algae, are the sites of photosynthesis, and this process in chloroplasts converts solar energy to chemical energy by absorbing sunlight and using it to drive the synthesis of organic compounds such as sugars from carbon dioxide and water. In addition to having related functions, mitochondria and chloroplasts share similar evolutionary origins, which we'll discuss later. The evolutionary origin of the peroxisome, as well as its relations to other organelles, is still a matter of some debate. So let's have a look at the evolutionary origins of mitochondria and chloroplasts. Now, mitochondria and chloroplasts display similarities with bacteria that led to the endosymbiont theory, which you can see on your screen. Now, this theory states that an early ancestor of eukaryotic cells engulfed an oxygen-using non-photosynthetic prokaryotic cell. Eventually, the engulfed cell formed a relationship with the host cell in which it was enclosed, becoming an endosymbiont, a cell living within another cell. Indeed, over the course of evolution, the host cell and its endosymbiont merged into a single organism, a eukaryotic cell with a mitochondrion. At least one of these cells may have then taken up a photosynthetic prokaryote, becoming the ancestor of eukaryotic cells that contain chloroplasts. This is now a widely accepted theory, which we will discuss. Now, the endosymbiont theory is consistent with many structural features of mitochondria and chloroplasts. First, rather than being bounded by a single membrane like organelles of the endomembrane system, mitochondria and chloroplasts have two membranes surrounding them, so they have two each. Chloroplasts also have an internal system of membranous sacs. Second, like prokaryotes, mitochondria and chloroplasts contain ribosomes, as well as circular DNA molecules like bacterial chromosomes associated with the inner membranes. The DNA in these organelles programs the synthesis of some organelle proteins on ribosomes that have been synthesized and assembled there as well. Third, also consistent with the probable evolutionary origins of cells, mitochondria and chloroplasts are autonomous, somewhat independent organelles that grow and reproduce within the cell. So let's have a look at mitochondria and how they're involved in chemical energy conversion. Now, mitochondria are found in nearly all eukaryotic cells, including those of plants, animals, fungi, and most unicellular eukaryotes. Some cells have a large single mitochondrion, but often a cell has hundreds or even thousands of mitochondria. The number correlates with the cell's level of metabolic activity. Now, this is important. For example, cells that move or contract have proportionally more mitochondria per volume than less active cells. Each of the two membranes, including the mitochondrion, is a phospholipid bilayer with a unique collection of embedded proteins. You can see that on your screen. The outer membrane is smooth, but the inner membrane is convoluted with infoldings called cristae. Now, the inner membrane divides the mitochondrion into two internal compartments. The first is the intermembrane space, the narrow region between the inner and outer membranes, and the second compartment, the mitochondrial matrix, is enclosed by the inner membrane. The matrix contains many different enzymes, as well as the mitochondrial DNA and ribosomes. Enzymes in the matrix catalyze some of the steps of cellular respiration. Other proteins that function in respiration, including the enzymes that make ATP, are built into the inner membrane. As highly folded surfaces, the cristae give the inner mitochondrial membrane a large surface area, thus enhancing the productivity of cellular respiration. This is another example of structure-fitting function. Mitochondria are generally in the range of 1 to 10 micrometers long. So let's have a look at chloroplasts and how they capture light energy. Now, chloroplasts contain the green pigment chlorophyll, along with enzymes and other molecules that function in photosynthetic production of sugars. Now, these lens shaped organelles about 3 to 6 micrometers in length are found in leaves and other green organs of plants and in algae. Now, the contents of a chloroplast are partitioned from the cytosol by an envelope containing two membranes separated by a very narrow intermembrane space. Inside the chloroplast is another membranous system in the form of flattened, interconnected sacs called thylakoids. In some regions, the thylakoids are stacked like poker chips. Each stack is called a granum. The fluid inside the thylakoids is the stroma, which contains the chloroplast DNA and ribosomes as well as many enzymes. The membranes of the chloroplast divide the chloroplast space into three compartments: the intermembrane space, the stroma, and the thylakoid space. This compartmental organization enables the chloroplast to convert light energy to chemical energy during photosynthesis. As with mitochondria, the static and rigid appearance of chloroplasts in micrographs or schematic diagrams cannot accurately depict their dynamic behavior in a living cell. Their shape is changeable, and they grow and occasionally pinch in two, reproducing themselves. They are mobile, and with mitochondria and other organelles, move around the cell along tracks of the cytoskeleton. The chloroplast is a specialized member of a family of closely related plant organelles called plastids. One type of plastid, the amyloplast, is a colorless organelle that stores starch, amylose, particularly in roots and tubers. Another is the chromoplast, which has pigments that give fruits and flowers their orange and yellow hues. So let's have a look at the peroxisomes and oxidation. The peroxisome is a specialized metabolic compartment bounded by a single membrane, and you can see that from your screen. Now, peroxisomes contain enzymes that remove hydrogen atoms from various substrates and transfer them to oxygen, producing hydrogen peroxide. This is a byproduct. Now, these reactions have many different functions. Some peroxisomes use oxygen to break down fatty acids into smaller molecules that are transported to mitochondria and used as fuel for cellular respiration. Peroxisomes in the liver detoxify alcohol and other harmful compounds by transferring hydrogen from the poisonous compounds to oxygen. H2O2 formed by peroxisomes is itself toxic, but the organelle also contains an enzyme that converts H2O2 to water. This is an excellent example of how the cell's compartmental structure is crucial to its functions. The enzymes that produce H2O2 and those that dispose of this toxic compound are sequestered away from other cellular components that could be damaged. Specialized peroxisomes called glycoxysomes are found in the fat-storing tissues of plant seeds. These organelles contain enzymes that initiate the conversion of fatty acids to sugar, which the emerging seedling uses as a source of energy and carbon until it can produce its own sugar by photosynthesis. How peroxisomes are related to other organelles is still an open question. They grow larger by incorporating proteins made in the cytosol and ER, as well as lipids made in the ER and within the peroxisome itself. Peroxisomes may increase in number by splitting in two when they reach a certain size, sparking the suggestion of an endosymbiotic evolutionary origin, but others argue against this scenario. Discussion of this issue is ongoing.